The purpose of this segment is to give us an overview of some of the structural features of proteins. Because as we go through the semester of chemical biology, what we are going to see over and over again is that the structure of the protein is going to determine the function of the protein and the types of applications that we can get for that protein in the field of chemical biology. And so we need to understand the groundwork and the fundamentals of protein structure. And how this is going to work is we can break protein structure into 4 levels of organization, starting with a primary structure that is the simplest and working our way up to the quaternary structure, the most complex level of organization. So let's break it down and look at those different levels of organization, including understanding of the intramolecular interactions that go on to enable this level of organization to occur within proteins and ultimately confer the wide array of functions that proteins have as structural stabilizers of cells, as enzymes that catalyze chemical reactions, and as a whole array of other essential functions. So thinking about first and foremost, when we are looking at the amino acids that are linked together via those peptide bonds that we talked about in the last segment, we need to consider the structural constraints that are put into place by the fact that we have those bonds present there, the peptide bonds. And so what I'm going to do is I'm going to go ahead and get the screen share up here so that we can take a look at what's happening. And the handout that I am using here that you may wish to print and scribble on your e-tablet or print out and scribble on old school style is found under modules for module 1, and it's going to be called something about protein structure. So this is going to be the handout we'll look at here in detail. So first things first, when we're thinking about the structure of proteins, we need to think about what are the restraints on the structures of the individual amino acids that form those peptide bonds, and what parts of those structures can we rotate around to make different conformations and ultimately assemble through intramolecular interactions into the overall complex 3-dimensional shape of a particular protein. So focusing on the peptide bonds, those are common to all of the links that join the amino acids, unlike the R groups, which are the variable parts of the protein. When we think about the peptide bonds, what we are going to notice is that the peptide link can undergo resonance, and that's going to have important implications for the structure or the confinement of the structure of the protein. It's going to impact what the protein shape is because the resonance is going to impact the allowable rotation of the peptide bond. So taking a look at specifically what I'm talking about here, when we talk about the resonance of the peptide bond, scribbling this here at the top of my slide. We're looking at the resonance of a peptide bond. If we draw out our prototypical peptide bond here for a generic peptide, we'll just work on a dipeptide here. So we would have a squiggly line here to represent the rest of that second amino acid that forms this peptide bond. Squiggly line here on the left to represent the first amino acid that is involved in forming the peptide bond. And then we have the central component here, outlined with my laser pointer, the peptide bond itself. And so what's going to happen, I'll go ahead and fill in my lone pair electrons to make it really obvious, hopefully what we're doing with resonance here, is that by resonance, we can create a second resonance structure where we can take the lone pair of electrons from the nitrogen, bring those down to make a carbon-nitrogen double bond, and in order to do that, to avoid Gouverneur octet rule, we need the pi bond electrons to come up onto the oxygen. And so if we draw out the resulting resonance structure that would result there, again, putting squiggly line in to represent the rest of the amino acid on the left, we would have a oxygen anion here with 3 sets of lone pair electrons squeezed in there. We would have, connecting the carbon to the nitrogen, a double bond there. So I'm putting the pi bond in there in red for emphasis. We'd have our NH group here, and then coming over, I'm just going to put a squiggly line, and remember that positive formal charge on the nitrogen. So if we think about this resonance that's going on here. What the resonance is going to do is it is going to result in all three of the atoms of the system, that is the nitrogen, the carbon, and the oxygen, here, here, and here, all behaving as trigonal planar sp2 atoms. And sp2 trigonal planar atoms are not going to have free rotation as a result of this resonance going on to put a double bond here in one of the hybrid structures and the double bond here in the other hybrid structure. So as a consequence of this being trigonal planar due to the resonance that's going on there, what we are going to see is that we will have this portion of each peptide bond, shown here and here, be locked into that trigonal planar conformation. It's going to be locked into that structure due to the resonance that's going on here. So there is not going to be free rotation around any of these highlighted bonds between the carbon and oxygen or the carbon and nitrogen because of the resonance that's going on there. And so those are locked in this trigonal planar conformation. And so when we think about what aspects of the protein structure are able to rotate around and engage in interactions with other groups within the protein, it's going to be the R groups that are going to be our groups that are capable of rotating around and forming different conformations so that they can, by rotating around to different conformations, engage in such things as hydrogen bonding and other interactions with other portions of the protein to allow the protein to fold up into elaborate three-dimensional shapes. So as we think about our protein structure here, if we come over to say this part and we were to go ahead and draw in our alpha carbon here with the hydrogen and the R group bonded there, it's going to be this portion of each amino acid that comprises the protein that we are able to have rotation occur of these bonds because we don't typically have resonance at this location. And so we'd be able to get rotation around our carbon-carbon bonds that are occurring here. And that rotation is going to result in a variety of different possible conformations being the three-dimensional shapes of that portion of the molecule. And we're going to have some very favorable conformations, three-dimensional aspects there, that will result in very specific defined structures overall of proteins. And we're going to see some themes that recur when we look at what types of structural elements these amino acid chains will fold into. And so we can organize that information about the structure of a protein into pretty much 4 different hierarchical levels. So we'll start with the primary structure. The primary structure we have already talked about in the last video. We didn't mention it too specifically, but that's just the sequence of amino acids that is linked by peptide bonds. So you can give all the information you need to know about the primary sequence of a protein by just listing out your string of amino acids that form a peptide. So just making a random string of amino acids here to form my peptide. I could keep going and going and going on there. That would be our primary structure. So primary structure is just what amino acids are linked to one another. It doesn't tell you anything about the three-dimensional shape of the protein overall. It's just who's linked to whom. Remember, we're always starting, as we said in the last video, with the N-terminus at the left end, as I'm showing with the laser pointer here, and the far right end, which I guess I'll put over here since I put ... there, would be the carboxy terminus, the C-terminus. So then from there, in order for the protein to actually have a biological function, for example, as a catalyst for a chemical reaction, it needs to organize into a very specific, well-defined three-dimensional shape. And the start of that organization is the secondary structure, which we describe as a transient three-dimensional structural shape that is formed by the protein when the protein is in solution. And so when a protein is thrown into solution, it will start to fold up into some common three-dimensional elements. And the two three-dimensional elements that you should be familiar with as we think about secondary structure are the so-called alpha helix, shown here as I'm highlighting in blue, and the beta sheet. So the alpha helix looks like an old school phone cord if you've seen telephones from the 1980s or early 90s. And what is going to be the case with the alpha helix is that, interestingly, the helical shape, the corkscrew shape that this forms, is going to be a right-handed corkscrew. Now, it's not theoretically impossible that you could have a corkscrew that is coiled the other direction, left-handed coiled instead of right-handed coiled, but that is not the typical paradigm. Typically, it's going to be a right-handed alpha helix. The other common theme that we see in terms of secondary structure, in terms of these transient three-dimensional elements that we will see, is referred to as a beta sheet. That's what we see here on the left middle side here is our beta sheet. And the beta sheet has a sheet-like structure that we represent by these arrows illustrating the orientation of those beta sheets. The beta sheets will tend to lay side by side by side, forming a sheet, kind of like a sheet on the bed lays flat. And so what is going to happen in both cases, in the case of the alpha helix and the beta sheet, is that hydrogen bonding is the key to forming these structures. And depending upon the particular amino acids that you have in the protein, you're going to get different patterns of hydrogen bond formation to yield these alpha helices or beta sheets. So what holds the alpha helix or beta sheet together is they're going to be held together formed by hydrogen bonds. Remember that hydrogen bonds are strong interactions, not covalent bonds, not ionic bonds. They're not really bonds at all per se, but instead they're really strong interactive forces between atoms that are positively polarized, specifically hydrogen atoms that are bonded to oxygen or other highly electronegative atom, such as a nitrogen. So you'll have a really strongly polarized, positively polarized hydrogen bonded to a negatively polarized atom, such as a nitrogen or oxygen. That's going to create strong positive polarization on the hydrogen. And then the other player in a hydrogen bond is some other atom, such as an oxygen, that is very negatively polarized elsewhere within the molecule. So you have a negatively polarized oxygen bonded to a positively polarized carbon, and we get a strong interaction between those groups. That strong interaction I'm going to put as a dotted line here, indicating a hydrogen bond. It's a strong attraction between those two groups. And it just so happens that the amino acids that make up the peptide chain just so happen to have a variety of carbonyl groups, because a carbonyl group is one of the groups that makes that peptide bond, and a variety of nitrogen atoms as well. And so we get hydrogen bond networks going on to yield an alpha helix or a beta sheet, depending upon exactly which atoms engage in these hydrogen bonds. And so we can take a look at that as we come down the page here. On the right, we have our alpha helical structure, and we can point that out as this helical shape that's going on up the chain. And our hydrogen bonds here are represented as the red dotted lines, which I'll highlight here. You can notice that the oxygen atom, which would be strongly negatively polarized, is engaging in a hydrogen bond, specifically with the hydrogen atom from another amino acid. And those amino acids are not adjacent to one another, they are further down the chain here. Likewise, we have a carbonyl group oxygen here engaging in a hydrogen bond with the hydrogen from a nitrogen there. Same theme here and same theme here. And that hydrogen bond network is what is creating this alpha helix. That is what's helping to organize the structure into this alpha helix. And we will typically see a whole network of alpha helices present within a particular protein. So you generally won't just see one alpha helix if you're looking at the diagram for a protein. Instead, you'll see a whole network of these alpha helices side by side. Generally, though this representation that we see here on the screen is a bit simplified and short, generally a typical alpha helix is usually about 35 amino acids in length for this single alpha helix. And then you'll generally see that this theme often repeats. You'll have a bunch of 35 amino acid alpha helices all lined up within a protein. So then what's going to happen is that you can also have a variety of minor modifications in this theme to get structures that are alpha helix-like, but maybe not completely identical to what we have drawn out as a schematic here. Then the beta sheet is an alternative way that hydrogen bonds form to start organizing the structure into this beta sheet as another type of secondary structure. And what's going to happen is that in this case, the hydrogen bonds are going to stabilize the structure by forming this sheet-like formation. And I will highlight this as well. These red dotted lines are what are denoting the beta sheet. I'm highlighting one in blue here on the left middle of your screen, another one here, and so on and so forth, going through this network where we have a variety of hydrogen bonds that are helping different parts of the molecule. You can think of them as the hydrogen bonds help the different parts of the molecule to stick together so that it assembles into this beta sheet type structure. So that's our secondary structure of the protein. It's when we're getting these sort of transient structures, transient three-dimensional elements that are parts and pieces of the protein as a whole. Then we need to jump up to the next level of complexity, which is what we refer to as our tertiary structure, going onward in this handout. And the tertiary structure is going to represent the interaction of multiple secondary structural elements to stabilize the polypeptide structure into a complete single polypeptide. And so when we talk about the multiple secondary structural elements coming together, we're talking about multiple beta sheets, multiple alpha helices, all of the elements that come in to stabilize and organize the structure of a polypeptide into a complex three-dimensional shape. And so looking at this structure that we see here on the screen, this is a tertiary structure of an enzyme called carbonic anhydrase. And what carbonic anhydrase does is it catalyzes the reaction of carbon dioxide and water to give a compound called bicarbonate, which is essential in the blood for maintaining the blood pH. And so this particular enzyme is upregulated or downregulated in a cell based on the needs of the cell for bicarbonate. And looking at this structure, what we can see, thinking about the tertiary structure of this protein in particular, is that we have a whole network of beta sheets here, which I'm circling in blue. And that network of beta sheets, which we've described as the secondary structure of the protein, comes into play with some alpha helices that we see in pink, a variety of alpha helices, and some other less organized, more random coil parts of the protein, such as what we see up here, what we see over here, are other structural elements. So, and I should add that although I use the term random coil, there really isn't randomness so much to this as when we say random coil, we mean that it doesn't fit into the theme of an alpha helix, a beta sheet, or some other common structural element. So therefore we call it random, even though it is generally very specific to that particular protein and oftentimes does play a critical role in determining the function of the protein. So the tertiary structure is the interaction of multiple secondary structures, multiple alpha helices, beta sheets, and other types of structural elements to stabilize the polypeptide structure. And this tertiary structure is going to be what's formed by the entire single peptide structure. And in many cases, that single individual peptide structure, that whole chain of amino acids that's organized into a three-dimensional shape, will have a very specific biological function in and of itself. It will be able to work as a catalyst for a chemical reaction, or it will stabilize the structure of a cell, or transport a particular ion from inside the cell to outside the cell or elsewhere. And so tertiary structure is sometimes the end point of structural organization of a protein, that when the protein reaches the tertiary structure, it is ready to go. It is ready to perform its biological function, such as catalyzing the formation of bicarbonates. On the other hand, there are some proteins that need to organize into an even more elaborate structure. And in those cases, the next level of organization is the so-called quaternary structure. And the quaternary structure is when you have multiple peptides, multiple polypeptides interacting with one another. So for example, in the case of this potassium channel, which transports potassium in cells, we have 4 different color-coded chains, which I'm highlighting here in pink. 1, 2, 3, 4. And those 4 different structures there represent 4 different polypeptide chains. So this is really 4 different proteins that associate together to work toward a common function, serving as a potassium channel in this case. So this is 4 different polypeptides. And those polypeptides are interacting through intermolecular interactions, such as hydrogen bonds. They are not covalently bonded to one another. In other words, there are going to be separate peptide bonds within the protein in purple than the protein that's in pink, orange, or green. So we have 4 different polypeptide chains. This structure here, that's a quaternary structure, has 4 polypeptides, would correspond to 4 totally separate primary sequences, where each of these individual so-called subunits that come together to make up this full functional structure, each of those would be encoded by a different gene, giving a different polypeptide, and then those 4 different polypeptides in this case work collaboratively, and they interact strongly to associate with one another into this quaternary structure. So a quaternary structure you can think of as multiple polypeptides coming together to work together as a single entity. And each of those polypeptides is referred to as a subunit. So we'll be using that terminology later on in the semester when we look at proteins in a bit more detail, referring to ones that have multiple polypeptides coming together to work collaboratively. So what we are going to do then in continuing to look at our biomolecules is we will next be getting into other classes of biomolecules, such as carbohydrates coming next in the pipeline, then lipids and nucleic acids, to further tie together all of this information that we need as our background for chemical biology.